All right, welcome to the second part of the urinary system screencast. Believe it or not, I think the first part was the hard part, and it'll be a little bit more straightforward moving forward. I just wanted to recap. So where we had finished, we were talking about filtration and spent a lot of time talking about all the mechanisms that affect glomerular filtration rate, because that's the easiest way to affect how much urine you're producing and all that stuff. But where we are in terms of how your kidneys are processing things is here. I just wanted to review what's in the filtrate, right? So everything in the plasma smaller than a protein leaks into the filtrate. This means lots of water, which your body would like to hang on to. Stuff that you want, other additional things that you want to hang on to, like glucose, vitamins, sodium, and stuff that you don't want, like urea and other waste products. So your kidney has to do a lot of sorting of what you want, what you don't want, and get as much stuff as possible into the urine with as little water as possible. So now we are here. We're going to briefly talk about reabsorption and secretion in general, and then what we're going to do is go through the rest of the nephron and talk about what's reabsorbed or what's secreted in the different regions of the nephron. So first thing, and this we're just talking about this, most of the glucose, amino acids, and vitamins in your urine are going to be reabsorbed, along with most of your sodium. Not necessarily all of your other ions, but your body does like to hang on to sodium. And then one of the things your kidney is going to do is balance how many hydrogen ions and how many bicarbonate ions end up in the urine through both reabsorption and secretion, so that you can balance the pH of your blood. And we're not going to worry too much about what happens where when it comes to pH control. We just want to know the end results, what's going to end up in the urine, so that the blood maintains the ions it needs to achieve the proper pH. And what you want to remember about sodium reabsorption is, again, it is our friend, the sodium potassium ATPase, that is doing it. This is why you can hang on to all of your sodium, but you can't hang on to all of your potassium. Looking at this, the one thing you want to remember is that water absorption is tied to ion absorption. So when you pump solutes like glucose and sodium out of the filtrate, you're leaving a lot of water behind and concentrating solutes in the bloodstream. This creates an osmotic gradient, so there's more water in the filtrate than there is in your bloodstream, and water is going to flow by osmosis from the filtrate into your bloodstream. There are channels or pores in the tubule cells that encourage water to flow through them, called aquaporins, so that the osmotic reabsorption of water is very efficient. When it comes to secretion, for us, we really just want to think about this middle bullet point here, is that this is your body's attempt to move things against their concentration gradient into the urine, so that if you're using a pump, you can pack more waste products into the urine than would enter either by filtration or by diffusion. So now what we're going to do is go through each part of the nephron and simply talk about what happens in each part of the nephron. First up is our proximal convoluted tubule. That is right here. As the arrow up there indicates, there are a lot of things that are reabsorbed in the proximal convoluted tubule. So the bulk of solute reabsorption, vitamins, glucose, amino acids, all of that, that takes place in the proximal convoluted tubule. So because that's where most of the solutes are reabsorbed, that's also where most of the water is reabsorbed. I'm going to say this a bunch of times because at the end we're going to talk about water coming out of the collecting duct, and it's going to make it sound like most of the water comes out in the collecting duct. But it is the last little bit of water that you can get out to make concentrated urine. But if you recall, oh, we don't have time to go into it, and I don't remember the term, so we'll just keep moving. This stuff also tends to run at a relatively constant rate. So your proximal convoluted tubule just goes on autopilot, and then glomerular filtration rate is altered or kept constant to accommodate these processes here. So now we're going to move from proximal convoluted tubule down into the descending limb of the loop of Henle. And all we want to know about the descending limb is that water is reabsorbed here. So this water coming out is going to go into a blood vessel and get carried away. Then we go down the other side, up. Now we are in the ascending limb of the loop of Henle, and we want to know in the ascending limb, ions, primarily sodium, is pumped out of the filtrate. And then we move up into distal convoluted tubule, and we are going to think of distal convoluted tubule as the part of the nephron that is hormone sensitive, that sort of runs at higher and lower speeds. So this is where aldosterone will cause increased sodium reabsorption. And what aldosterone is doing is just causing those tubule cells to manufacture and insert into their membrane more sodium pumps. So you just have more pumps all working at a constant rate, pumping more sodium out of the filtrate so that water follows. And this is also where parathyroid hormone acts, causing calcium to be reabsorbed from the filtrate back into the bloodstream when your blood calcium levels are low. Now we are on to the collecting duct. Again, this is where the final concentration of filtrate takes place. Off the top of my head, I want to say your glomeruli collectively produce somewhere around 70 liters of filtrate a day. You produce 1 to 2 liters of urine a day. So 68 liters of filtrate. Right, let's say you're secreting 2 liters of urine a day. Out of that 70, that means 68 liters are reabsorbed in your proximal convoluted tubule and loop and distal convoluted tubule. So the overwhelming majority of your water that is reabsorbed before the water gets into the collecting duct. But if you are slightly dehydrated and you only want to produce 1 liter of urine versus 2 liters of urine, that last extra liter, right, which represents 50% of the water that's left in here, that gets pulled out in the collecting duct. So because there's very little water left here, if you take more water out, it makes a big change in the concentration, but it's not a large proportion of water. So this is caused or regulated by antidiuretic hormone, which you remember comes from the posterior pituitary. It is going to cause the insertion of aquaporins into the collecting duct, which is going to enhance the reabsorption of water through the collecting duct. We are going to come back to that in just a minute, but I have a little detour. I think in the future I'm going to move this slide, but it's too late for today. And we're going to talk about pH. Again, we're not going to worry about exactly what happens where in terms of what is reabsorbed, what is secreted, and in what part of the nephron. We just want to know how is the kidney going to change the contents of the urine so that the contents of the bloodstream are shifted towards a better pH balance. So let's assume now that your blood pH is too high. Your blood is alkaline. Do you have too many or too few hydrogen ions in your bloodstream? You should be thinking too few, right? So high pH means low hydrogen ion concentrations. So you want more hydrogen ions in your bloodstream, which means less of them are going to end up in the urine. At the same time, if you are too alkaline, right, you don't want a lot of bicarbonate in your bloodstream, because bicarbonate that's your alkaline reserve. It can absorb hydrogen ions. So you're going to ensure that you have more bicarbonate stays in the filtrate or ends up in the filtrate, fewer hydrogen ions, so then in the bloodstream you've got less bicarbonate and more hydrogen ions. So conversely, if your blood pH is too low, does this mean you have too few or too many hydrogen ions? Right. Now it means you have too many. So pH goes up, pH goes down. If it's acidic, that means there's more hydrogen ions. So now you want to dump all those hydrogen ions into the urine, because we're talking about what ends up in the urine there. And then, conversely, you want fewer bicarbonate ions in your urine. You're going to leave them in the bloodstream so that they can neutralize the excess hydrogen ions that you have. So that's regulation of pH, and just know what ends up in the urine under these two conditions, and don't worry about exactly how the kidney achieves that. So now we have to get back to the collecting duct, because right now we're talking about how are the events in the collecting duct regulated, and why is it that the collecting duct is so good at pulling a little bit more water out of your urine. The reason why the collecting duct is good at pulling water out of the urine is because you have what is called a medullary osmotic gradient. What this means is that if you're out here in the cortex, there is not as much salt, among other things. So the osmolarity, or total solute concentration, of that environment is relatively low compared to the inside of the medulla, or as you go sort of deeper into the medulla, it gets more and more saltier. There are more and more solutes. So the osmolarity increases as you go from the cortex through the medulla. This is going to play a role in concentrating the urine. So how does your collecting duct and this osmotic gradient, how do they combine to concentrate your urine? So first things first, if you're making dilute urine, then all of that doesn't matter. There isn't, or at least there are low amounts of antidiuretic hormone. So whatever the osmolarity of the filtrate is when it enters the collecting duct, it's pretty much the same when it leaves the collecting duct. However, if you're dehydrated and your posterior pituitary is releasing antidiuretic hormone is going to cause, what are they called, aquaporins, to be inserted into the membrane here. That is going to make the collecting duct more permeable to water. So now what you have is water flowing through an increasingly salty environment. So up here, there's some salt, more salt here than there is in the filtrate in the collecting duct, so that pulls some more water out. That concentrates your filtrate. Now your filtrate is more concentrated, but it finds itself in a region of the medulla which also has a higher concentration of salts, so more water continues to come out. So as the filtrate gets more and more concentrated, it's traveling through a saltier and saltier environment, so water keeps coming out, and that's how your kidney is able to squeeze the last few drops of water out of your urine that it can get out. Your urine is still like 95% or something water, but there's a lot less of it in dilute urine, in concentrated urine versus dilute urine. So now there are two things that we need to understand about this osmotic gradient. One is, how does all of that salt get there, or what mechanism establishes the gradient? and you are going to see that as our countercurrent multiplier. And then why doesn't the sodium just float away? And what is preventing all of this water that's leaking out of your collecting duct from diluting all of the salt that's been dumped into your medulla? So we have to answer the two questions: how does all the salt get there, and why doesn't it just diffuse away? And the answer to those two questions are the countercurrent multiplier and the countercurrent exchanger. The mechanisms by which these two phenomenon act are a little complicated. What you need to know is simple, and I will point it out as we go. So first things first, how does all of that salt get there? It gets there because of the countercurrent multiplier, which is the loop of Henle, and that is what establishes the gradient. So those are the two facts you need to know. The loop of Henle is the countercurrent multiplier, and that establishes your medullary osmotic gradient. That is why there is so much salt in the medulla. So the way it works, and it works this way again because of this countercurrent. You're flowing down here and up here through the same environment. It's a little easier to understand if you start in the ascending limb. So in the ascending limb, it's impermeable to water, but salts are pumped out. That makes your medulla a little salty. So when more filtrate flows through this end, that pulls water out. If you pull water out, your osmolarity goes up, right? You're concentrating the solutes in the filtrate now. So when filtrate enters, it's 300 milliosmoles or 300 milliosmoles of salt per liter of filtrate. And by the time you get down here, it's 1200 milliosmoles of total solute, I should say, in every liter of filtrate. So if you're concentrating the solutes, that makes your pumps more efficient. So think about if you're bobbing for apples, or if there's just a whole bunch of apples and a barrel full of water and you're trying to scoop out as many as possible. If there are a whole ton of apples floating on the surface of the water, you're going to be able to scoop up bunches of apples at the same time. But if there's a relatively small number of apples in a very big barrel of water, you're going to have to hunt around and find them all to pick them out, and you're not going to be as efficient as picking them out. So basically what you're doing here, if we're using our apples in a barrel analogy, is that you're draining a lot of the water out of the barrel so it's easy to find all of the apples. It's one way to think about it. So that is the loop of Henle, which is your countercurrent multiplier, and it answers the question, why is the medulla so salty? Or how does it get so salty? Then we have to talk about what's the other question? Why doesn't the salt just diffuse away? And the answer to that question is, I don't know why I'm making these changes because I'm not going to save it. The countercurrent exchanger, i.e. the VASA recta. So you need to know that the VASA recta is the countercurrent exchanger, and it is the mechanism that is responsible for maintaining the osmotic gradient. This one to me is even a little bit more complicated. I don't think I fully understand it, but here's how I think about it and how you should think about it. You have blood, it's going to flow down this side of the VASA recta, turn around, and then flow back up this side. As the blood flows down, sodium comes into the blood and is carried down into the medulla, and then as the blood comes up, the sodium goes out and the water goes in and the water is carried away. So it's always exchanging sodium and water as it's going down and up. And the way I think of it is that it's carrying the sodium down, dropping off the sodium, picking up the water, and then carrying the water away. So what it effectively does is pushes the sodium back down in and literally carries the water away. There has to be a mechanism to get rid of the water, and the best I can understand is that that's what your VASA recta does. So those are two complicated phenomenon, but your understanding of them only has to be very simple. Now we are on to, this is just the odds and ends at the end of the chapter, so we'll go relatively quickly. You simply want to know that a diuretic is anything that enhances urinary output. And I promise you I will not ask any questions about the difference between an osmotic diuretic or some other kind of diuretic, but it is fun to think about different mechanisms by which different kinds of substances can increase your urinary output. This slide, not really important at all. I am not going to ask you what your urine looks or smells like, because I don't want to know. This one, kind of important. I simply want you to remember that pH is slightly acidic. I will not ask you for a number range. I simply want you to remember that it is slightly acidic. This is going to come up again when we do the male reproductive system because urine and sperm share the same exit port. They both leave via the urethra, so there has to be a mechanism to neutralize any leftover urine in the urethra before sperm pass through there so they don't die on the way out. Chemical composition of urine, not really important. What I want you to remember and think about is that the most prominent waste product in urine is your nitrogenous wastes. So urea, uric acid, and creatinine. Creatinine comes from your muscles. It's a breakdown product of creatine phosphate, if you remember that. And I think I said in the first screencast, urea, uric acid, and ammonia are all nitrogen-containing compounds. This is because all proteins have amino acids in them, and all amino acids contain nitrogen. If you are eating protein, but not using the protein that you eat to build your own proteins, like you eat a steak, but you're not working out, you're simply burning that steak for energy. All of those amino acids have to have that amino group with the sodium in it clipped off before they can be shuttled into your carbohydrate respiratory metabolism. And you've got to get rid of that nitrogen because there's no other use for it in the body. Moving right along, kidney stones, they suck. I'm probably not going to ask about them, but I hear they're really unpleasant. Let's move on. This will slow down on a little bit. There's 2 slides left, but there's probably 1 or 2 questions on these 2 slides, so it's worth studying, mostly because it's just really easy to ask questions about this. What does it mean if you have albumin in your urine? Albumin is... I can't remember the word, and I did this. This always happens to me with albumin. It is the most prominent or popular protein in your bloodstream. So you have more albumin than any other protein in your bloodstream. So we tend to think of that being the protein that shows up in your urine. What this means is that you've damaged your glomerulus, right? Most likely your basement membrane, because that's the part of your filtration membrane that is supposed to restrict proteins, from entering your filtrate. Glucose is also not supposed to be there. This most usually means you've got diabetes, right? And it doesn't matter whether it's type 1 or type 2. Either lack of insulin or insulin resistance both means insulin is going to stay in your bloodstream. It's not going to get absorbed into your tissues. So when you make filtrate from your plasma, there's going to be more glucose than usual in the filtrate. More glucose than usual is going to end up in the urine. That also pulls water along with it, which is why you have polyuria or increased urinary output when you have diabetes. That would be an example of what do they call it? One of those diuretics. Now I can't even remember. An osmotic diuretic, glucose in a diabetic patient. Last up, red blood cells. If there are holes in your glomerulus big enough for a red blood cell to get through, you've got serious damage to your kidney. It could also mean an infection. I'm not really sure how that affects the kidney. I'm going to assume all sorts of inflammation in your kidney, causing your glomeruli to be ridiculously permeable to the point where even red blood cells can squeeze through. And blood disease here, I think that refers to if you have hemoglobin or some remnants of red blood cells showing up in your urine. That can happen if you have lots of red blood cells dying. White blood cells, that generally means you've got a UTI, an infection somewhere in your urinary tract. Most likely your ureters or urethra or bladder, and then white blood cells by diapedesis are going to crawl into your ureters or your urinary tract and help fight the infection. If there are microbes there, that most likely means you have a UTI. Urine, generally speaking, is clean or sterile. So as long as the urine doesn't come into contact with any body parts before it gets collected, it generally doesn't have any bacteria or viruses in it, and it's usually not considered, for this reason, a biohazard, so you can just toss urine samples down the sink. Ketone bodies, as this says right here, are a product of fat catabolism, which means fat breakdown. So for whatever reason, if your body is primarily relying on fats for energy instead of carbohydrates or proteins, then you're going to end up with ketone bodies in your urine. So you might be digesting fat because you're starving and your body is tapping your long-term fat stores. You could be on a low-carb diet, like the relatively extreme ketogenic diet, which I haven't really heard good things about in terms of its effects on your health, but I know some people who swear by it and they lose weight on it. But that diet is a very high-fat, very low-carb diet, and it just doesn't sound healthy, but apparently people swear by it and they lose weight. So you'll leave a good-looking corpse when you die of kidney failure. That's probably hyperbole, but whatever. And then diabetes, right? So if you have diabetes, you've got plenty of glucose in your bloodstream, but it's not getting into your cells, so your cells are all acting as if there isn't any glucose in your body, and they're breaking down fats and proteins instead of the glucose, which they can't get a hold of, and you end up with ketone bodies in your urine. That is the end of the digestive system. Thank you for hanging in this long. We'll get together again. What's next? Reproductive. Woohoo!